All right, it says noon on my clock. So, hello everyone. Welcome to Grand Rounds and Happy New Year. It's January 9th. I think I can still say Happy New Year for another couple days. So, welcome. This is our first Grand Rounds of 2026. So, thanks for being here. We've got a great talk. Um, but before we get started, I do want to kind of go over some of the logistics here. I want you to have the best experience possible with today's grand rounds. So let's talk about some of the things you need to know. First thing to know, we are recording this. So if you didn't catch the whole thing, or if you want to watch it again later, we'll have a recording posted in our Grand Rounds archive sometime next week. So you can find that through our website. We have a huge archive. There's about 10 years worth of Grand Rounds in there. So take a look at that sometime if you have a few minutes to spare. Let's see. We are going to try to leave a little bit of time for questions at the end of the presentation. I'll return to kind of moderate that section. So if you have anything, go ahead and use that Q&A feature down there at the bottom of the webinar. If you type your question into the chat, I'll see that too. I'll be paying attention, but we prefer that Q&A feature. So go ahead, type those questions in whenever you think of something. We'll get those queued up, and I'll come back on at the end of the presentation, and we'll get those questions answered before we wrap up at 1 o'clock. Okay, what else? There's also a survey that'll show up at the end of the webinar. As soon as it closes, you'll see a link to that survey. And if you want to give us any feedback about today's presentation or suggestions for future presentations, we'd love to hear it. Go ahead and enter that in the survey at the end. And I wanted to let you know that we will not have a grand rounds next week because of the holiday weekend, but then we will return on January 23rd with Dr. Patricia Arion, who will be giving a talk called Services and Interventions Research in the Context of Mental Health in the USA, a 5-year perspective. So mark your calendars for that one, January 23rd. All right, so if we want to go to the next slide here, I think that's probably enough for me. So I'm now going to toss it over to Dr. Susan Ferguson, and she is going to introduce today's speaker. So I'll disappear. Over to you now, Susan.
Great, thanks, Jed. And yes, I'm super pleased to introduce our speaker today, Kevin Coffey, who'll be giving a seminar entitled Of Mice and Misuse, Preclinical Modeling of Fentanyl Use Disorder. Kevin is a research assistant professor in psychiatry and behavioral sciences here at the University of Washington. He's also a MERIC investigator over at the VA Puget Sound. Kevin did his undergraduate, master's, and PhD training in psychology all at Rutgers University. And then following that, he joined John Neumeyer's lab here to conduct his postdoctoral training before starting as faculty in 2023. Kevin's lab aims to identify the neural mechanisms underlying substance use disorders and maladaptive decision making. His lab's current focus is on studying the neurobiological consequences and predictors of chronic fentanyl use. And to accomplish this, his lab utilizes cutting edge in vivo optical neuroscience tools like photometry, optogenetics, and miniscopes, along with the newly developed oral fentanyl self-administration model for rats and mice. He's also the lead developer for DeepSqueak, which is a popular software package for bioacoustics analysis that integrates AI algorithms with an intuitive graphical interface to accelerate animal communication research. And in Kevin's seminar today, he will review the use of rodents to dissect the neurobiology of fentanyl use disorder, focusing on work that his lab has been doing around preclinical modeling using oral drug self-administration. And so with that, I will turn things over to Kevin.
All right. Thank you, Susan, very much for that introduction. Like you said, I'm Kevin. I started my lab here at UW and at the VA just 2 years ago, but I've been part of the psychiatry research community for the better part of 10 years now. So it's really exciting to finally get to give this talk. Like Susan said, our lab is focused on the neurobiology of fentanyl use at the moment, and I'll jump right into it. Okay, so why fentanyl? I don't think I really need to explain this to most people in this seminar, but fentanyl has become a really significant public health crisis in the last decade. And it is a synthetic opioid that is very cheap, it is readily available, and it is extremely potent. And for a long time, it was primarily an adulterant in other opioids, but in this day and age, it has become a drug of choice for a lot of users, particularly the most severe opioid users, young people, and people with comorbid psychiatric conditions. And because of all this, fentanyl is responsible for the majority of opioid overdose deaths and drug overdose deaths. I used to update this figure every year or so, and the line basically just kept heading up off the page. Fortunately, in the last year or so, thanks to a lot of hard work in public policy and in medical treatment. I think a lot of people that come to this talk have been involved in providing medicated assisted treatment, that sort of thing. The fentanyl overdose death rate has actually plateaued and I believe started to come down in the last year or so, which is really exciting. Um, but that said, there is still a very visible and significant public health crisis going on. And so, as a basic behavioral neuroscientist, we still want to do our part and help and see if we can't you know, learn more about what goes on in the brain and hopefully improve treatment. So, like I said, what can we do as basic neuroscientists? So, the work to address the fentanyl crisis is multidisciplinary and is going to involve massive societal change and healthcare and housing. But as a basic neuroscientist, there are things that hopefully we can do to make this easier. So my lab kind of has three approach vectors that we're working on right now when trying to address the fentanyl crisis. The first is a very simple question. How does fentanyl change the brain? This is the kind of classic approach for neuroscientists. This is your brain on drugs kind of approach. For a long time, this was focused on small circuits and individual genes, things that were kind of like reductive. But what we're trying to do is take a bigger whole brain approach, an approach that looks at kind of the brain collectively and holistically. We're also really interested in individual differences and susceptibility, since severe and problematic substance use seems to be limited to a pretty small subset of the population, and even those that are regular users of opioids. The next thing we're focused on in the lab, this is a smaller part of the lab, it's something that we're trying to expand, is the ability to test novel therapeutics. So we have a recent ADA pilot grant where we're looking at the use of GLP-1 receptor agonists. These are drugs like Ozempic, semaglutide, that seem to reduce more than just appetitive eating, but other intake of other substances as well. And then finally, a big chunk of the lab is focused on negative reinforcement in addiction. So, like I mentioned before, the kind of like bulk of addiction research for a long time was looking at brain areas that are positive affective reward centers. So how does the brain hijack, or excuse me, how do drugs hijack the reward system? But they're also really strong negative reinforcers in addiction. So individuals report not wanting to quit because withdrawal is so severe. Stress is a common way to induce relapse. And there are also people taking consistently despite negative consequences. So there's a lot of negative reinforcement mechanisms, and these are mechanisms that we think are essential to target for treatment, since withdrawal avoidance and relapse are such serious deterrents to prolonged voluntary abstinence. Okay, so the first question, how does fentanyl affect the whole brain? So about 2 years ago, we received a large multi-PI R01 to ask this question, how does the fentanyl affect the whole brain? The idea here was to collect researchers with different kind of modern toolkits for looking at the brain and ask questions about what fentanyl does to it. So this is a collaboration with researchers in the Department of Neurobiology and Sorry, it used to be biological structure. It just changed. I think it's neurobiology and biophysics now, something like that. Anyways, Lin Kai Gu, who's there, is the developer of PixelSeq. This is a spatial transcriptomics technique. He's able to map gene changes in tissue across large swaths of tissue at one time, which is a really exciting, highly specialized technique. There's Sam Golden, who is an expert in clearing brains. So this means actually taking whole brains and turning them see-through so that he can image them all at once in intact and look for areas that are active during particular behavioral points that are interesting to us. And then there's Nick Steinmetz, who is the developer of NeuroPixels. These are very high density electrophysiological probes that can record from thousands of neurons at the same time across vastly different brain regions, so brain regions that are far apart, looking at how they communicate. These techniques are all really amazing, but what they needed to be able to apply them to fentanyl was a good ethologically relevant model of fentanyl use, one that could capture the different components of fentanyl addiction that we're interested in studying, so that we can ask, interesting biological questions using these really modern new techniques. So for the last year, we've been developing a new model for mice. The kind of gold standard self-administration models are called IV or intravenous self-administration models. These are really commonly used because they work very well. Basically, you put an indwelling catheter into the animal and they can take drugs directly into the bloodstream. These work very well, but they have some issues. They are kind of complicated. There is the risk of surgery and attrition. And when you're combining this with these other complicated neurophysiological techniques, there's just a lot of risk points in the line where you could ruin your experiment. And so what we wanted to do was develop a convenient and robust, easier to develop model of oral fentanyl self-administration. And fortunately, fentanyl is extremely potent. And so you can mix fentanyl in with water, the animals can drink it and get the kind of doses that are necessary for good biological effects without the aversion of drinking like a salty water because it's so potent. So on the left, you'll see our brand new setup. What we focused on here is modular, expandable, scalable setups. These are actually just cheap Suncast plastic cabinets that can each house 3 different running environments. They're hooked up with low-cost camera systems and audio recording, all of which uses open source software to analyze. And we're giving away kind of the plans for this along with the publication so that this can be replicated in other labs. What does this look like? So here is an animal that's trained to press a lever. They get a little light cue and then go immediately over to this hopper and drink their fentanyl. You'll notice their body is being covered in a little skeleton there. We're also developing a open source tracking model for these exact chambers so we can release that as well. And researchers can get really high resolution information about the position and movement of animals during the different periods of time during the study. And we're still working on refining it for things like when they're in the hopper and most of their body is obscured. Okay, so why is this self-administration model useful? So what we're trying to do is cover as many of the different components of addiction in humans that we can with these models. So hopefully you can see my mouse. During self-administration, these animals escalate intake, so they take more over days. This is common in human addiction. The second phase we have here called extinction models involuntary abstinence, and we can measure how much animals seek, even though there's no drug available, kind of persistence in seeking drug. And then we have relapse tests. These are often done with exposure to cues, such as the context or other cues associated with the drug, or sometimes by exposing the animal to stress and measuring how much they seek drug under those conditions. So we have a really good coverage for the different epochs in addiction we're interested in these animals. And then I also mentioned we're interested in individual differences. So in animal models, the kind of type of animal you use matters. We classically use these black mice up top, these C57s. They're an inbred strain that's got very consistent behavior, but we also ran this with an animal called the CD1 mouse, who is an outbred strain. And while their behavior doesn't seem to mimic the human condition as much, they are highly variable. And so they can be a valuable tool when looking at, say, individuals that take a lot or very little when we want to separate those kind of high risk and low risk animals. And then the last thing I'll say about the behavior for this model is that you really need to prove that the animals are, doing this behavior because of the drug, because they actually are trying to take fentanyl. And there's lots of ways you can do that, but one clever way is to, unbeknownst to the animal, change the dose. So these are animals that are used to taking a particular dose of fentanyl, and if we suddenly start to lower the dose, you'll see the animals will start to increase their responding. And that's because they're trying to maintain the intake that they prefer to intake. And so they're good at doing that at the higher doses. And as the dose gets lower and lower, they actually struggle to respond enough to keep it up to where they want, but they are basically trying to. So they modulate seeking proportional to the dose. And the CD1 mice are less capable of doing this, but to an extent they do it too. Basically, they start to increase responding as the dose drops. And then finally, borrowing a technique from Susan. Thanks again, Susan, for that introduction. We are able to separate these animals into kind of risk severity categories. So we can take all of the behavior data that we've collected and kind of combine the individual elements that are related to addiction, like total intake, the amount of seeking, the association between the cue and the reward, relapse responding, that sort of thing, and create a composite severity score so that when we go to study the brain, we can ask questions about the individuals that are high and low risk, what makes them different, what brain areas are active. And just showing that in another way, you can also take all this information and kind of project it out into severity space with something like a PCA. The takeaway here is just that certain animals, you can see kind of what defines their high risk trajectory. So our CD1 animals have a lot of extinction responding and relapse responding. Our C57 animals are more associated with escalation of intake, lots of seeking, and total intake, that sort of thing. Okay, so what's going on in the brain of these animals? So this is all really new data. This is the first of the kind of 3 bits of that collaboration that we've collected data for. Sam's lab, Lucas McMillan and Todd Appleby, 2 really excellent young scientists, have been taking these brains and clearing them. So what that involves is a step called delipidation, actually pulling the fats out of the tissue and then impregnating the tissue with an optical clearing media. And then with a special microscope that fires laser sheets through the brain, we can image it while it's intact from the top to the bottom and isolate cells that are active at the period of time just before the brain was removed. And doing this, we can map out in 3-dimensional space what brain areas are overactive at very particular moments in time. And so I'm just going to show one piece of the data that they produced recently. So this is looking at the relapse test and then a control just afterward. And on the brain sections on the left here, you'll just notice that there are a lot of brain areas that are active after exposure to drug cues and a relapse test that are kind of spread all throughout the brain. So this really reinforces the idea that single brain areas responsible for say reward systems, things like that, are not the only things changing. This is a holistic change in the brain, and the goal in the future is to take these individual animals with all their individual differences and see if we can map on changes in brain areas, correlate basically the differences in activity in different areas with the risk for certain behaviors. So say risk for high relapse rates. That is all an ongoing process. Basically right now we're just doing the looking at 2 groups, basically right after a relapse test or a control group. And like I said, we see really, really large changes in areas like the cortex, striatum, pallidum, areas that are involved in emotion and cognition and memory, not just basic reward centers. All right. Like I said, the next thing that we're interested in looking at is treatment of fentanyl use disorder with novel therapeutics. Thanks to the Alcohol and Drug Abuse Institute here at UW, we had a pilot grant recently to look at the use of these GLP-1 receptor agonists for treatment of fentanyl use disorder. And so I want to just point out that we already have good treatments. Medicated assisted treatment for fentanyl use is, I think, by and far away the most effective treatment we have right now. There's a lot of people here at UW that are responsible for helping promote the use of buprenorphine and other medications for addiction. There are some issues with these drugs. They are essentially opioid agonists, they're replacement therapies, and there are a lot of myths surrounding them. So obviously, these drugs are a lot better than street drugs. They have much longer half-life, so they don't cause constant kind of ups and downs, cycles of withdrawal. They're administered in a way that's safe and effective. They don't cost your whole salary to get them, and so they're very, very good. But there are some inconveniences and people that due to myths and stigma don't want to use them, and so it's always good to have another tool in the toolkit. That's where GLP-1 receptor agonists have come in. So you might be familiar with them from weight loss and from diabetes management. They are drugs that reduce intake of food through acting on kind of like the gut. It turns out some of them cross the blood-brain barrier and seem to work on the motivational systems in the brain. And so they are thought to reduce other appetitive behaviors like alcohol intake. There's some evidence in nicotine, and recently there's been some evidence in opioids. And a big benefit of these drugs is that, one, they're already approved, which means they can go into the hands of people very quickly. And two, they're very conveniently administered. So they can be administered at home. They're administered weekly for weight loss. And if they were able to be administered weekly for addiction, that would be great. And so that's basically what we've set out to test with our animal models. For this, we're using rats, and basically what we've done is modify the self-administration paradigm so that the animals first take 2 weeks of fentanyl. So they're currently on fentanyl. That's the population we'd like to treat with these, not prevention. So you want to be able to give this to someone who's currently on fentanyl, and then we start giving them slightly higher doses each week and look if that changes their behavior. So this was really promising. But basically, what happens is that the two groups, the dulaglutide, that's a particular GLP-1 receptor agonist that we're studying because it crosses the blood-brain barrier well, both animals increase intake over 2 weeks. And then when you start to give them the dulaglutide, they basically level off. We don't see evidence yet that they will quit taking fentanyl, but the non-dulaglutide animals will continue to escalate. They'll take more, quite a bit more over time. And so even that is pretty valuable to prevent escalation. We have a lot of behavioral data from these animals, so we know everything about weight change and vocalizations that they're making. We're still working on analyzing that as well as the data from the brains of these animals. It's basically just a long process of getting in line and having the labor and students to do it. But we're very excited to hopefully turn this into an R01. We're in talks with collaborations to maybe run this kind of experiment with novel GLP-1 receptor agonists, ones that are more targeted at addiction than, say, weight loss. So that's all coming down the pipeline. For now, it was just a really nice pilot. Okay, and then the bulk of the lab. So a big chunk of the lab is focused on negative reinforcement in fentanyl addiction. I like to show this figure a lot in talks. I think it really gets across some of the points that I'm going to talk about. So addiction is often seen as this cycle of first the positive rush of intake. People take drugs because they do enjoy that rush, but very quickly drug levels fall. They go into withdrawal. It's unpleasant, and over time, kind of the highs start to fade and the lows get bigger. And then during, say, a period of a voluntary abstinence, life stressors come back, and they really, people are sensitive to that stress. They have very deep negative affective states, often called like hyperkatifia, sensitivity to negative reinforcement and negative affective states, and it promotes continued drug taking. And while a lot of previous research, like I said, is focused on the positive mental state side of this, the hijacking of reward systems, we're really interested in brain regions that are processing the negative reinforcement side. So things like, why is withdrawal feeling so severe? Why do people avoid that? Why do stressors induce relapse? That sort of thing. And so I will bring up my lab's favorite brain region here first, the lateral habenula. So the lateral habenula is a really ancient brain structure, and it's conserved across like almost all vertebrate species. So it exists in lampreys and bony fish and all the way up through mammals to humans with very similar input-output structures. And the lateral habenula sits at kind of this crossroads, the gateway between forebrain structures, which process emotion, positive emotions, as well as negative emotions, and then midbrain structures, which are responsible for motivating behavior. And so it's a really key node in processing aversive events and motivating behavior. And generally, it's seen as very adaptive to have this structure that's existed for millions of years. It has to have good evolutionary advantage. But when things go wrong, it's also associated with anxiety disorders, major depressive disorders, addiction, that sort of thing. But yeah, like I said, it exists for adaptive reasons. It just as often goes wrong in disease. Okay, so how do we model some of these things? This is going to be a slightly different model than self-administration. We are really interested in withdrawal avoidance. So this is, why do people not want to withdraw? It's because it feels terrible. Even the memory of withdrawal is terrible. For opioids, it's a pretty severe physical ailment, as well as then sensitivity to emotional distress. For these kind of experiments, we use something called conditioned place aversion. This is where animals get injections of fentanyl over a period of time, and then we confine them to a particular context while they're on fentanyl. Then we can give them something like naloxone, which is a drug that will, one, prevent overdose, but in these animals, it causes instant and severe withdrawal. And during that time, we can confine them to another context. And then on a final day, if we put them back into a chamber and say, which context do you want to spend your time in? Generally, they avoid the context where they went through withdrawal. And then we can ask questions about what's going on in the brain when they're exposed to that context, what goes on in the brain when they're avoiding that context. So just to give you a little idea of what that behavior looks like I said, the animals spend a lot less time in that chamber that was associated with naloxone or withdrawal. They do, however, spend some time in there, and they travel back and forth between the two chambers. And so we can look at brain activity when the animals are in that chamber or when they're making transitions. As Susan mentioned, one of the techniques that we use to look at brain activity during these behaviors is called fiber photometry. It's a technique where we can genetically modify the cells in the brain to actually glow when there's activity in them. And then we can use a glass fiber and a photosensor to record the changes in brain activity while the animal is behaving. And when we did this during the conditioned place aversion test, what we noticed is that when the animal makes a transition from one side of the chamber to the other, we get a big increase in activity. And that's interesting, but it's not that interesting since it happens whether they're moving from the withdrawal side to the saline paired side or the saline paired side to the withdrawal paired side, right? It's not necessarily related to the aversive chamber. But the very dedicated undergrad who was video scoring all of these things noticed that these animals also make a lot of halted transitions. So they kind of make a beeline for the door between the two chambers and then make rapid stops. And What she noticed is that when the animals make a rapid stop before entering the naloxone paired side of the chamber, we get a really strong spike in lateral habenula activity. And we don't get that when they make a rapid stop before entering the saline paired side of the chamber. So we really do think that the habenula is involved here in that decision to not enter the withdrawal paired area. But the habenula is a very complicated area. It's very small, and this technique really only lets us look at global signal. So, what are all the cells doing? So, in the future, we're moving towards new techniques that I'll talk about, where we can look at kind of individual cells that their activity may be changing in different directions and masking the kind of true activity of the area. That makes sense. Okay. And then we have a lot of work, like I said, in oral fentanyl self-administration. I'm not going to go over this again just because you already saw it previously, but we do this in rats to study negative reinforcement as well. And one thing that's really useful about rats is that they are highly communicative. So they make something called ultrasonic vocalizations. These are social communication signals. They're vocal signals that are well above the human hearing range. And they basically sound like bird calls or whistles if you slow them down enough that we can hear them. Most of these calls are used in social contexts, positive contexts, when they get sugar water, or they're engaged in sexual activity, or social play. But there's a particular subset of calls called long 22 kilohertz vocalizations that are used more as alarm calls or stress calls. So they'll even make these calls when they're alone, and they're almost always associated with negative affective states. And so if we record these calls during fentanyl self-administration, we notice something kind of interesting. So animals like to self-administer in a very particular way. They make a lot of responses early in a session to raise their drug level up to a certain point, and then they transition to making slow and spaced out responses so that they can maintain a particular drug level. And we used to call this drug level satiety, or the point at which the animal was satisfied. That's where they want to stay, akin to eating food until you're satiated. But if you look at the vocalizations the animals make during this, they make a lot of the positive affective calls during the first 15 minutes during the rush. That's when the drug level is rising rapidly. But while the animals are maintaining a steady state drug level, they're actually only making the negative affective USVs, a lot of these negative calls. And so what we think is happening is that every time the drug level dips down, the animal feels bad and is going back to the lever to try to alleviate that negative affective state. So this is akin to, over time, the positive rush going away, but we're seeing this really, granularly within session. The animals are right away feeling that negative state and trying to avoid it. So we're really interested, again, what's going on in the brain while this is happening. We have done a lot of recordings of the lateral habenula, like I said, and we noticed that the lateral habenula is always processing drug-related cues, so the cues that are associated with fentanyl, but it is highly sensitive to the animal's internal affective state. So the cue processing during this maintenance phase, when the animal is ostensibly in a negative affective state, is really heightened. So we think what's happening is the habenula is, modulating, learning about the cues based on the animal being in this negative affective state. What's also interesting is what the animal takes the drug when they consume fentanyl. We see the signal in habenula drop really fast. So we have a signal that's very high when the animal is hearing a cue and seeking, and then dropping again once the animal takes the drug. Okay. So what do we do with this information? So I've talked a lot about measuring the brain and figuring out what's going on. That's kind of the first step of a lot of our projects. The second step is to manipulate the brain, to use techniques to change that activity and see if we can change behavior. And so these experiments are usually a little harder, but we have a big project going on right now. Our postdoc Sierra has been working very hard to see if she can block this lateral habenula activity during fentanyl self-administration and see if it will change behavior. And basically what she's doing is genetically modifying the cells in the lateral habenula to be sensitive to a light source. And she's using short pulses of light to basically inactivate those cells at the period of time when we think they're contributing to fentanyl seeking. And so this is a pretty large and ongoing project. She is getting close, I think, to being able to present this data, and hopefully there's a conference that a lot of the addiction researchers here at UW are going to called the SPEAR Conference. Sierra should be taking some of this data there and get to share it. But for now, I'll just touch on it. Okay. And then the last project I want to talk about, yeah, we got good time, is fentanyl seeking despite negative consequences. So one thing that can be kind of hard to model in the lab is the fact that animals will continue to take drug despite humans will continue to take drug despite negative consequences. And humans, those consequences are really complex mix of, physical, social, emotional. There are really complex consequences for humans who are severe drug takers. But in the animal model, we can try to at least abstract that and look and see if there are correlates. So Ari Asarch, one of John Neumeier's grad students, has been working with me for the last year to modify the fentanyl self-administration model to include negative consequences. What this entails is training the animals to take fentanyl, oral fentanyl normally, and then in the final weeks of self-administration, we introduce blocks of negative consequences. What that means is that the animals get to take fentanyl during what are called safe trials, where a cue says, take fentanyl, nothing will happen. And then it switches over to a danger trial, where the cue signals that after they press the lever and get their fentanyl, they're going to get a shock delivered at some point in time. And those shocks increase in severity over days. And what we're interested in looking at here is where is the animal deciding to take drugs despite the negative consequence, and where is the animal deciding to stop because the consequence has gotten too severe. And so the behavior data here is pretty interesting. What Ari's found is that during the safe trials across the entire test, there really isn't much of a change. They know that the cues associated with safe trials mean they're going to get the drug and not get shocked. Once we introduce the shock, early on, the responding actually goes up. So very mild shocks seem to have a kind of motivating effect on taking drug. But then as the shocks get more severe, the total intake starts to drop until when the shock is quite severe, the animal basically takes no drug at all. And this is another way to show that. I just really like this figure. But to explain it, each of these tick marks is an individual response, and each row is an animal on the different shock days. And you'll see in those first 15 minutes here, the animals basically have the same pattern of intake the whole time. On the no shock days, they have this very distributed pattern. When the shocks start getting introduced, animals almost have these burst responses. I think they're trying to get in a lot of responding at once and only get one shock or something. Basically, it's really modulating their behavior. But by the high shock sessions, the animals basically don't respond at all. Ari also recorded ultrasonic vocalizations in these animals, and to no one's surprise, during the high shock session, these animals are producing a lot of negative 22 kilohertz USVs in that same period of time that they are in other sessions, but to a much greater extent. So these graphs have to be scaled to show exactly how much negative affect there is. So they really, really don't like having to choose between the drug and the shock. That's all we're showing here. Okay. So same thing, what goes on in the brain? We're still interested in the lateral habenula, but like I said, the issues with photometry that we have, where you're only recording all of the cells at once, you're getting like a global signal, they were giving us a lot of problems trying to determine if different cells are doing different things, if different inputs matter, how can we tell? And so what we've done is we've started to use a technique called miniscopes. This is a similar method where we have the cells genetically engineered to glow when they're active, but we actually put a full head-mounted microscope on the animal. This is a full fluorescence microscope, and then implant a optical lens into the brain. And these are capable of imaging individual cells deep in the brain during behavior. And so we can get an idea of what every individual cell under the lens is doing. This presents some serious technical challenges. We get a lot of imaging data, and from that have to extract all of the cells and their activity. Ari actually spent probably a year developing a brand new pipeline for this called the Miniscope Processing Suite. This paper is in review in eLife right now, and we're really excited to get it out to the field. Other labs have developed tools to do this, but they're often command line based and a little finicky, and kind of everyone's set up for their own particular lab. Ari has worked very hard to make this accessible with a graphic user interface, with tools to modify it to work for your lab, and a whole website and dedicated documentation, that sort of thing. But that's just a long story to say, when you have a whole lot of images of the brain and you have to extract the cells, now you can do that very easily. You might see them actually flashing down here on the bottom, but yes, we're going basically from thousands of pictures of the brain over many hours to traces of activity from individual cells. And from that, the idea is to look for different populations of cells that are active at different times. Okay, so this is all extremely new data, and Aria is working on it currently. Same thing, I believe. Hopefully, we'll be bringing this to the Spear conference. But what he's looking for right away is activity during the trial. Is it coming before the choice to hit the lever? Is it coming during the shock? That sort of thing. And what he notices early on in the early shock sessions is that the lateral habenula is not really responding to the trial cues, but it is responding to the shock. Basically, it's learning about the shock. Later on in the session, the cells seem to be responding to the period of time between a lever press and the shock, where the animal is anticipating that shock is going to come. And then later on, when the animals are well trained in this and they know that strong shocks are coming, the lateral habenula is actually responding to the danger cue. So before they press the lever, like they know that if they press the lever, that they're going to get shocked. It's like an anticipatory cue. These images I'm showing you, all of the rows in here are individual cells, and the activity at the bottom is averaged across them. But what Ari is working very hard on right now is a way to cluster the cells into individual communities, so ensembles that work together to process a particular behavior. And he's got a lot of data to sort through before we can isolate those. But this kind of data is the basis for some R01s that are going in right now in my lab and in John's lab to look at these individual ensembles and see if we can't manipulate them during behavior to change how animals are responding, change their risk tolerance, that sort of thing. Okay. I think that's pretty good for now. I am going to say a huge thank you to my lab. We are small but growing. Sierra, my postdoc, Jenna and Tam are techs, and Chao Yu, the undergrad. They're all great. And then my biggest collaborators right now, John and Sam, their labs have been really instrumental in helping me get off the ground. It's a tough time to be a new PI, but since the community here at UW is so excellent, I really been fortunate. And thanks to all the funding, and thank you, psychiatry. Questions?
Yes, questions. We've got plenty of time. So, folks at home, go ahead and type any questions you have in our Q&A feature down there, and I'll read those out loud. We don't have any that have come in yet, so let's give some people time to do that. I had one question, Kevin, a really basic one, while we're waiting for other folks to write some better questions. but mine is about the chambers, the different chambers that they're crossing between. What is different about those two chambers that they can tell the difference?
Yeah, so in those we just use visual cues. There are people that use other cues, so like odor cues. Those are a little harder to control, but people say like blind as a bat or mouse. The rats and mice actually have pretty good vision, and so high contrast visual cues are enough for them to know which chamber they're in.
Okay.
Yeah, if you want long-term learning of cues, odor cues are extremely potent. Rats and mice are just have incredible sense of smell.
Okay, interesting. Let me check the chat. I don't think there's anything in there either. Nope. Okay, so first question has come in. I'll read it out loud for the folks at home. So they say, potentially silly question. Are vocalizations to drug, specifically fentanyl, uh, conserved in humans?
Yeah, that is a great question. I don't know the answer to that. I do know that humans will self-report the rush, the positive affect during rush, and they will self-report the negative affect during the period of time when a drug is falling. So I think with humans, sometimes if you're not asking, you're not going to get the answer right. But these animals evolved in a way where they're in big groups. They're basically communicating, like yelling out to the ether, and so they'll do that even when they're alone in a chamber. And so I think the underlying concept is certainly conserved. Yeah, there are actually a lot of vocalization like conservation between humans and rats in other conditions too. So like we have some collaborations where we're looking at infant rats that have, like in human conditions like autism have vocal disturbances. The rats will also have vocal disturbances in hypoxic ischemia, things like that. So there's a lot of overlap between human vocalization issues and rodent vocalization issues.
Great. So our next person is asking, um, so who engineers the equipment that can make all of these intricate measurements?
Yeah, so we have a mix of handmade equipment and commercial equipment. For a long time, there's like one company in like behavior space called Med Associates. We use a lot of their equipment, but these days with 3D printers and small micro computers like Arduinos and things like that, we do a lot of it on our own now. We also buy very inexpensive camera systems that we integrate with a lot of incredible open source software to do behavioral tracking. So all of the camera like footage you see are special infrared like single board cameras that cost less than $30. But the footage from them we can put into open source behavior tracking models and get highly detailed, highly accurate behaviors. And Sam Golden's lab has been instrumental in writing some of the software to do behavioral classification. Our lab developed, like I said, Deep Squeak, which does the audio classification.
Okay, great. Um, so can you say more about the undergrad's observation of the pauses? So how much time were they spending with the animals and or the data?
Yeah, so that project was done before we had really good video tracking, and so sometimes it's let's say worth the time of an undergrad to sit and watch like dozens and dozens of hours. They usually watch these videos at like 2 times real time and are just sitting there marking exact times when they make chamber transitions. So when you watch dozens of hours of rats, you start to notice some of their stereotype behaviors. And yeah, rats have something called approach avoidance behavior. So they're naturally kind of nervous and curious, both of those things. And so they do these things where they'll dart towards somewhere and like stop very rapidly. Like they're like there's something going on where they've made that decision. And so that's what gave us the idea to look in the brain at that precise decision point and see if there was any differences.
Yeah. Okay, so next question. Curious if you're examining individual differences, such as high and low responders, in assessing resilience to the drug.
Yes, absolutely. So that is a main focus of that first grant. We have a really good method of kind of putting the animals into different risk and resilience categories, even down to the level of which types of behaviors they're most at risk for, like, being the really high intake animals or really high risk for relapse. Um, and Susan has also done, like I said, a lot of that work in her lab. The issue there is that it takes a lot of animals to have very good statistical confidence in the like comparison between, a high risk animal and their brain activity and a low risk animal and their brain activity. And so those are basically always like the step after we compare the big group differences. And so this project, being relatively new, we're just collecting as much data as possible. And yeah, I think we're probably, within the next 6 months, able to do that with the tissue clearing data. And then hopefully the next step is that spatial transcriptomics I was talking about. We're getting as many brains to them as possible. Unfortunately, you can only use one brain per animal per assay for these things. So it just takes a long time to collect that kind of data, but we're definitely doing it.
Okay, well, you say, I mean, so a lot of this is really new, but the next question here we have is where do you see this research? Where do you hope this will be in 10 years?
Yeah, I mean, I think the goal is, precision treatment. I think some of it's predicting who will be high risk and resilient. I really want to go down that route, but it's very difficult work. But some people are looking at, if we can predict beforehand which people are at highest risk, we can, prevention is always better than treatment, right? Um, I think that's going to be a big area of research. But then we already have a ton of people who need treatment who are severe addicts right now. So just knowing how to treat different people who are at risk for either overdose or risk for stress-induced relapse or really sensitive to cues, being able to provide information to treat those individuals more effectively. That's the goal. That's the goal.
Yeah. You talked a little bit about how they like to maintain a certain drug level. How do you know? How do they know like when to stop? Like what tells them something how much is too much?
Yeah, so one thing that's kind of different about these animal models than the human condition is that the animal knows, they're very good learners. They're very good at associating. They know how big the dose is each time. And so over a period of a couple weeks, they can feel in your body like how how much drug is in there. And we actually have modeling techniques where we can model the kind of brain concentration of drug. And animals that are good at this maintain like very, very tight drug concentrations, especially with intravenous self-administration because the feeling is so immediate. And then we've done studies where you can artificially clamp the drug level just above or just below where the animal is like prefer it. We call preferred, but now we know they're just trying to escape negative consequences. And if you clamp it just even 5 or 10% above what their level that they like is, they'll stop responding entirely. They'll just sit there and take it. If you clamp it 5 or 10% below in an area that they find aversive, they will respond relentlessly. We have a paper from cocaine days years ago where we had animals responding thousands and thousands of times over the course of a couple hours because they really are trying to escape that, like just too low feeling.
Yeah.
Very sensitive to the internal drug level.
Yeah. Okay. Well, the next question here is actually, do you collaborate with those who are trying to find vaccines for opioid use?
So uh, not yet, but we're working on it. Marco Pravatoni is also here in the psychiatry department, is looking at, well, obviously he has his whole vaccine development part of his lab, but he's also interested in developing new GLP-1 receptor agonists. So we've just started talking, fingers crossed that we can make something work where his lab is developing new drugs, and then we would be able to help out testing them.
Okay. Great. All right. It looks like we've gotten through all of our questions. So,
Great.
Dr. Coffey, yeah, thank you for being here today.
Yeah, thanks so much for having me.
Of course. Yeah. Great presentation. Good way to kick off the new year. And folks at home, yeah, thanks for being here. Please plan to join us on the 23rd. And have a good weekend, everybody.